Several firms now use microbes to produce high-value human milk oligosaccharides for infant formula. But could plants be a better production vehicle?
HMOs, or human milk oligosaccharides, are this really specialized fraction of carbohydrates, complex carbohydrates, that are present in breast milk. And in fact, there's a very unique collection of these molecules in human milk that aren't found anywhere else in nature. They're very important for programming the immune system early in life. They interact directly with the cells in the gut to reinforce that barrier, that reduce inflammation, adjust and then calibrate allergic responses. So very important biomolecules for early life development, which of course is why they're so sought in infant formula. It's one of the areas where it's one of the aspects of breast milk that is very difficult for bovine-based infant formula products to replicate. So, of course, parents want as close to breast milk as they can for their baby if breastfeeding is not available. So it's an important class of molecules.
You talked about, the hundreds of HMOs have been identified. Do we know which ones are important and, therefore which ones to focus on?
Yeah, it was really a surprise to me as I got into this field of the complexity of human milk, and to realize that so much about the structure and function relationships of the molecules within milk have not been mapped. And so HMOs, the R&D libraries of HMOs today maybe cover about 25% of the structures in human milk. So there's potentially 150 or more structures that we just have simply never been able to map what does it do into functional applications. So we'll be the first to be able to have a complete library to start to work with from that capacity.
So kind of currently a handful of these can be made or are being made through microbial fermentation.
Right. That's right. Yeah, there's a handful of them today that are made in genetically engineered E. coli. So they program the machinery for linking these sugars together into E. coli, and they can grow them in bioreactors. And this results in a pretty high cost of manufacturing and a pretty limited repertoire of molecules that are available. So human milk has hundreds of unique oligosaccharides in it, and only about 5 of those are available at scale today in a biomanufacturing technology.
Why might plants be a superior production platform for these complex carbohydrates versus microbes or animal cells?
When we were trying to come up with a chassis to make HMOs, we really saw plants as that natural chassis to make them because they're already so good at making sugars. Additionally, there is a huge ability to scale plants that really other systems can't match. You can grow plants at hectare scale, and arguably we do agriculture at the largest scale than any other technology in the world. So when you need to make a product at this huge scale so that you can make babies all over the world healthier and make adults healthier, we really see plants as the best platform for that, to scale it, lower the cost, and also expand the repertoire of HMOs that can be made with their mastery of sugar metabolism.
So what plants are you using as expression systems and why?
Yeah, so we kind of have 2 different strategies. The first one is our lab scale platform where we can test genes really rapidly. So one of the issues with plants is that it takes a long time to test things typically, but we have a system where we can test out different biosynthetic pathways in the lab in 3 to 5 days. And so it's really great for that initial discovery and proof of concept work. As we're going into real large-scale production, we're looking at plants that have already validated streams for making a variety of products. So our initial target is soybean because there's already existing infrastructure, and we think that it's going to provide a great amount of yield as well as a good diversity of structures.
So in the lab, would you then be using a kind of transient expression system with like a tobacco plant or something?
That's exactly right. Yeah, we're using Nicotiana benthamiana for our transient expression assays.
Okay, but that wouldn't be something you could scale for commercial production, or would it?
It depends on the product. I think we can see pathways either way for using it, either just as an R&D platform or for some specific molecules we could actually use it as a scalable platform to produce them at smaller amounts for specific use cases. We had been doing this work for 1 year with essentially no funding, and we were already able to make so much progress compared to the 30 years that people have been doing microbial fermentation of HMOs.
One of the advantages of your system, as I understand it, is you can tap into this infrastructure that's already there, that is easier to scale perhaps than precision fermentation. But there are also challenges as well. Can you monetize the other parts of the soybean? Do you have to set up expensive identity preserved supply chains? Can you speak a bit to those challenges?
Yeah, we certainly expect that it's going to be important to be able to trace the crop and the product all the way through that supply chain. And so, one of the advantages of working in soybean, as you said, is the existence of, kind of these closed systems that we'll need to be able to access and utilize in our process. So we need to design our business around, the systems that exist now and the ones that are starting to come online using some of the more advanced solutions for that kind of identity tracing. But the goal is to be able to monetize that entire bean, from the field into this high-value carbohydrate fraction at the end of the line. So we want to be able to sell the oil, we want to be able to sell a protein concentrate, whether that be for a human nutrition application or some other application, but hopefully to monetize the whole thing. The value proposition for the go-to-market strategy is that we'll be able to make the same HMO ingredients that are available today, but at lower cost.
Like the 2FL?
Yes, so we're focusing on 2FL, that's like one of the simplest HMOs, the easiest one to make, and it'll be the first in the food crop. And then we also believe we'll be able to make more complex structures, so it's really important that we go after one in particular called DSLNT that is out of reach for current technology, but has some really nice preclinical evidence behind it that it will be a life-saving solution for babies who are born too soon. So in the NICU, there's a large unmet need for this particular HMO, which has been shown to have a protective effect in that population. What we have learned is that they're actually really looking to expand HMOs into other age categories. So they really like the ingredients that they have available. They've done well with HMOs in premium infant formulas, but a lot of these companies have also funded clinical trials in other populations across the lifespan. So they want to see them in pediatric children's nutrition, as well as in food products and beverage products for all ages. And so they're really thinking about HMOs as part of their health and wellness specialized nutrition portfolios. They're also really frustrated with the high cost of HMO ingredients and have had a hard time justifying extending the molecules into those applications. So we're getting a lot of positive response to the possibility of a more affordable and accessible cost for those buyers.